Hello everyone. This is the first video in which I'm going to explain the first 5 questions of mock test 1 for the public channel. During the following 3 videos, we'll discuss the other 15 questions. It's possible for all students to attend this test. The deadline is just one day. It's just one day before the IMAT test. I will share the test link in the description. This test includes 20 questions from limited topics. Don't forget to subscribe our YouTube channel and like the video. So let's go and discuss about the question number 1. Water molecules are composed of 2 hydrogens and 1 oxygen atom. They are essential molecules in the human body because water could influence the body temperature. That's correct. It concentrates hypotonic solutions. I think we have to discuss more about hypotonic solutions, and this statement is wrong. It participates in many reactions, and it's correct. So the final answer is C, statement number 1 and statement number 3. But let's discuss more about water molecules. Water molecules are polar molecules. The bond between hydrogen and oxygen involves an equal sharing of electrons. So we can say the water molecule is polar. Each water molecule has 2 hydrogen atoms, which have a partial positive charge, and 1 oxygen, which has a partial negative charge. Water molecules have a bent shape, and 2 hydrogen atoms are on the same side of the molecules, and the oxygen forms the opposite poles. When water molecules are close together, the positively charged are attracted to the negatively charged of the other water molecules. The kind of attraction between water molecules is a kind of secondary bond known as hydrogen bond. We have 2 types of forces, primary and secondary. Primary forces are attractive forces between basic or fundamental particles like atoms and ions. Based on the type or types of particles involved, primary forces include metallic, ionic, and covalent bonds. Secondary forces or intermolecular forces, they have a different story. They are attractive forces between molecules in solid and liquid states. Let me repeat it again. Secondary or intermolecular forces are attractive forces between molecules in solid and liquid states. A hydrogen bond is a force that forms when a hydrogen atom in one water molecule is attracted to the oxygen atom of another water molecule. These bonds are weak individually, but their large numbers cause a significant total force keeping the molecule tightly together. Let's go and discuss about the functions. They have lots of functions in our body, and I prefer to mention 3 of them. Water molecules have essential roles in our lives, and they can be solvents in our body. Water can solve a great variety of inorganic and also organic molecules, creating a solution. Water molecules are attracted to the ions and polar molecules. It happens when a chemical substance dissolves in the solution. They form hydrogen bonds with polar molecules, right? And the best known example is cytoplasm. The cytoplasm is a complex. It's a complex of dissolved substances in which the chemical reactions of metabolism occur. But before going and discuss about the other function, I would like to mention 2 important words, hydrophilic and hydrophobic. The literal meaning of hydrophilic is water-loving. It describes polar molecules that react with water molecules and form hydrogen bonds. Examples include sugar and table salt, which dissolve in water. But it's very, very important. Not all substances in organisms are hydrophilic. Many many hydrophobic substances can be found within the cell. An example, fatty acids. The second function of the water molecules that I would like to mention is they can be an environment. Chemical reactions in the living cells happen in solutions. Water is the environment for many reactions, especially those which occur within the cell. And also, they can be a participant. Water molecules take part in many biochemical reactions. Later, probably we will talk about photosynthesis and also the formation of carbonic acid in the blood. So let's go and discuss about the second question. It's about the formation of one disaccharide molecule from two monomers. During the formation of maltose, two monomers are connected by central oxygen atom and one water molecule is produced. C. Before going and discuss about the monosaccharides, disaccharides, I would like to discuss about condensation and hydrolysis reactions. Condensation reaction is a reaction in which two or more molecules combine to form a larger molecule. It means that monomers will combine and at the end we'll have a large molecule. But it's very important to know that during condensation reactions, water molecules are produced. So products are large molecule and also water molecule. But hydrolysis, the story is totally different. Hydrolysis involves adding water to one large molecule to break it into multiple smaller molecules. Now we have a large molecule. We add water molecules to that and we can see new products. And the products are the monomers of that large molecule. So when we talk about the formation, the water molecule is produced. When we are talking about breaking the bonds between the monomers in one large molecule, the water is used. So it's very important. Maltose or malt sugar, it consists of two covalently linked alpha glucose units. We'll discuss about the alpha and beta glucose molecules, and the type of linkage is alpha 1 and 4. What does it mean? Look, it's carbon number 1, it's carbon number 2, 3, 4, 5, and 6. It's one glucose, right? And glucose is a monosaccharide and it has 6 carbon atoms. It has 6 carbons. It's connected to another glucose by the central oxygen atom. The name of this bond, it's glycosidic linkage. Is the kind of bond between two monomers. It's a covalent bond. And you can see here the type of the glucose is alpha. We have two types, alpha and beta. We'll talk. The type of the glucose is alpha, and it's from carbon number 1 to oxygen, and oxygen to carbon number 4. So it's about the direction. It's about the type of the glucose, and it's about the direction. And disaccharides are formed during condensation reactions and can be hydrolyzed by adding water to disaccharide molecules. And maltose is composed of 2 glucose, 2 alpha. Glucose has 2 isomers, and it depends on the orientation of the hydroxyl group. Look at here. When the hydroxyl group it has an opposite direction to the CH2OH group, the type of the glucose is alpha. But when it's in the same side as CH2OH, the type of the glucose is beta. So it's very important to know. Perfect. Let's go and discuss about question number 3. It's very important. I'm going through each row and we will discuss about them. The first one, the polysaccharide molecule is glycogen. What's the function of the glycogen? Glycogen is a storage polysaccharide in human body, in animals. It's correct, but can we find glycogen in all cells, like in brain cells, in the cells in our heart, or for example, the cells in the liver? We can find glycogen molecules only in liver cells and also muscle cells. So brain cells is incorrect. Is it a branched molecule? Yes. And the bonds are alpha 1 and 4, alpha 1 and 6. Right now, we know how to imagine alpha 1 and 4 in our mind. Alpha is the type of the glucose, and 1 and 4 is the direction. Alpha is the type, and 1 and 6 is the direction. Another polysaccharide is in a form of starch is amylopectin. Amylopectin is a branched molecule. So this no is incorrect. And it's a storage polysaccharide in plant cells. It's correct. But this no is incorrect. And the type of the bonds, alpha 1 and 4, alpha 1 and 6. Amylose, another form of starch. It can be found in plant cells. Storage polysaccharide in plant cells. Is the branched molecule? No. But the type of the bond is alpha 1 and 4. Cellulose. What's the function of the cellulose? Cellulose can be found in the cell wall of plant cells, not animal cells. Animal cells, they don't have cell wall. And the type of the bond is beta 1 and 4. And is it a branched molecule? No. So the correct answer is E. Let's discuss more about polysaccharides. A polysaccharide is a micromolecule consisting of repeating units of simple sugars, usually glucose. The polysaccharides are starch, glycogen, and cellulose. They might be a single chain or a branched chain. We have two types of polysaccharides, storage and structural. Storage polysaccharides like starch and glycogen, and cellulose is the structural polysaccharide. Let's discuss about each of them. Starch. Plants store starch, a polymer consisting of alpha glucose subunits. So the monomer is alpha glucose. It has two forms, amylose and amylopectin. Amylopectin is more common but has shorter chains than amylose, and it has side chains. It has two types of bonds, alpha 1 and 4, alpha 1 and 6. It's very important to know that alpha 1 and 4 represents the chain, the main chain of the amylopectin, and alpha 1 and 6, it represents the branched chain. So every time when you see alpha 1 and 6, it means branches. And when you see alpha 1 and 4, it means the main chain of the molecule. So imagine that you have no idea that we are going to discuss about amylopectin. And I mentioned that we have one polysaccharide, and the types of the bond is alpha 1 and 4, alpha 1 and 6. Is it a branched molecule or not? It's a branched molecule. And another form of the starch is amylose. Bonds are alpha 1 and 4. Is it a branched molecule or not? No, it's not a branched molecule. They have a storage. They are storage polysaccharides in plant cells, and most animals, include humans, they have enzymes to hydrolyze plant starch, and it's insoluble in water. Starch is insoluble in water solutions, and doesn't affect water potential. So it doesn't cause water to enter cells by osmosis, which would make them swell. So it's really good option to store in the plant cells. The second one is glycogen. Animals store polysaccharide called glycogen. It's a polymer of alpha glucose that it looks like amylopectin, but it's more branched. It's a highly branched molecule. And as I mentioned before, it can be found in liver and muscle cells. Bonds are alpha 1 and 4 and alpha 1 and 6. Wait. We have both alpha 1 and 4, alpha 1 and 6. Is it a branched molecule? Yes, it's a branched molecule. And two important hormones regulate the amount of sugar in my body, glucagon and insulin from the pancreas. And the third one is cellulose. Cellulose is the most abundant organic molecule on the planet. It's a structural carbohydrate. It contains beta-glucose monomers joined by beta 1 and 4 linkage. Cellulose is never branched, and some hydroxyl groups on its glucose monomers are free to make hydrogen bonds with hydroxyl of other glucose molecules, as you can see here. So it's never branched, but it's possible to see hydrogen bonds between the layers. And the human body doesn't have a specific enzyme to digest it. Question number 4. Choose the incorrect statement about the lipids. During the hydrolysis of fat molecules, 3 water molecules are produced. I think we discussed about it. We are going to discuss about hydrolysis, right? Hydrolysis. We have to add water molecule to the large molecule to have the monomers. So 3 water molecules are produced. Incorrect, produced, done. The first statement is incorrect. Unsaturated fatty acids tend to be liquid at room temperature. It's correct. The main component of the cell membrane is composed of 2 fatty acids. It's correct. Cholesterol is an essential structural component of the animal cell membrane. It's correct. And the oxygen atom or atoms can be found in the structure of lipids. Let's discuss about each option. Statement A. Fat or triacylglycerol is composed of 3 fatty acids and a glycerol molecule. 3 fatty acid molecules are each joined to glycerol by an ester linkage, a bond formed by a dehydration reaction between the hydroxyl group and the carboxyl group. The reaction between alcohols and the carboxylic molecules is esterification. You can see the ester bond here. And this reaction is esterification. 3 condensation reactions make a triacylglycerol or fat molecule. The first reaction yields a monoacylglycerol, and the second a diacylglycerol, which is an important molecule for sending signals within the cell, and the third a triacylglycerol. And also 3 water molecules are produced at the end. You have to know this reaction. Statement B. We are going to discuss about unsaturated fatty acids. We have 2 types of fatty acids, saturated and unsaturated. But wait, what's the difference between the saturated and unsaturated fatty acids? I think it's really obvious. Fatty acids with 1 or more double bonds are unsaturated fatty acids because they contain less hydrogen than they could. Unsaturated fatty acids tend to be liquid at room temperature. But why? Because the links where the double bonds are located prevent the molecules from packing together closely enough. So it's the reason that the Unsaturated fatty acids are liquid. They are not solid at room temperature. Statement C. The main component of the cell membrane is phospholipids. A phospholipid molecule consists of a glycerol molecule. It's the glycerol, and a glycerol molecule attached at one end to two fatty acids and the other end to a phosphate group. So one end to phosphate and another end to two fatty acids, or we can say hydrophobic tails. Phosphate has a negative charge, negative electrical charge in the cell, and phospholipids belong to a specific group of lipids called amphipathic lipids. The two ends of phospholipids show different behavior toward the water molecules because phospholipid molecules have both hydrophilic head and also hydrophobic tails. So we can study more about phospholipid molecules and also their functions in cell membrane, right? Cool. Statement D, we are going to discuss about cholesterol molecule. It's a type of steroid. It's very, very important molecule in animals and a common component of animal cell membranes. So till now, we discussed about two lipids, phospholipids and cholesterol molecules, and we can find them within the structure of the cell membranes. Steroids are lipids characterized by a carbon skeleton consisting of 1, 2, 3, 4 fused carbon rings, and different steroids have different side chains. So we are going to discuss about cholesterol so you can see the side chains, and other steroid molecules have different side chains, and their functions depend mainly on the different functional groups attached. And the last question is question number 5. It's very, very important. We're going to discuss about proteins. Find a mismatch. Beta-plated secondary structure. You have two forms, beta-plated and alpha helix. Hydrogen bonds mention the alpha helix structure. Both are correct. A sequence of amino acids. We're talking about primary structure. Am I right? Perfect. So it's correct. Hemoglobin. Okay. Two polypeptide chains and it has a quaternary structure. Hemoglobin has a quaternary structure. It's correct, but two polypeptide chains? No. Hemoglobin has 4 polypeptide chains, and myoglobin, it has 1 polypeptide chain and has a tertiary structure. It's correct. So D is the answer, but let's go and discuss about all types of proteins and also a little bit about hemoglobin. 4 main levels of protein organization. Primary, you can see, secondary, tertiary, quaternary. Let's go. Primary proteins. A polypeptide or protein molecule may contain several hundred amino acids linked into a long chain. So primary structure of a protein is the sequence of amino acids. So we just discussed about the sequence of amino acids. For example, glucagon, a hormone secreted by the pancreas, is a small polypeptide with only 29 amino acids. So what's the key word from the primary proteins? A sequence of amino acids. Secondary proteins. Most proteins have segments of their polypeptide chains repeatedly coiled or folded in patterns that contribute to their overall shape. The coils and folds collectively referred to as secondary structures, result from hydrogen bonds between the repeating components of the polypeptide backbone, you can see here. There are two basic shapes that the polypeptide chain can take. The first one is alpha helix. Alpha helix looks like a telephone wire, and beta plated, which looks like a folded piece of paper. Perfect. And tertiary proteins. It's very important. It's very, very important. While secondary structure involves interactions between backbone components, tertiary structure is the overall shape of a polypeptide resulting from interactions between the side chains of the various amino acids. The three-dimensional structure is determined by four main factors that involve interactions among R groups belonging to the same polypeptide chain. These include both weak interactions and strong covalent bonds. So, first one is hydrogen bonds. They form between R groups of specific amino acid subunits. The second one, an ionic bond, can occur between an R group with a unit of positive charge and one with a unit of negative charge. Hydrophobic interactions result from the tendency of nonpolar R groups to be excluded by surrounding water and therefore to associate with the interior of the globular proteins or globular structure. And the last one is covalent bonds, are known as disulfide bonds, or sometimes they mention disulfide bridges. It's a kind of a link between two sulfur atoms, and it's very, very specific. It's for a cysteine amino acid. It's very, very important to know that tertiary proteins, they have only one polypeptide chain. And one of the most important examples is myoglobin. And the last one is quaternary structure. Many proteins consist of two or more polypeptide chains. Quaternary structure is the overall protein structure that results from the aggregation of these polypeptide subunits. The same types of interactions that produce secondary and tertiary structures also contribute to quaternary structure. These include hydrogen bonding, ionic bonding, hydrophobic interactions, and disulfide bridges or covalent bonds. One important thing that I would like to mention about disulfide bridges is that they are a common feature of antibodies and other proteins secreted from the cells. Other examples of quaternary proteins are collagen and hemoglobin. The shape of the hemoglobin is spherical. The four polypeptide chains pack closely together, and the hydrophobic R groups pointing towards the molecule center, and the hydrophilic ones pointing outwards. So, right now we know that hemoglobin, it can be found in the red blood cells. They carry oxygen and also carbon dioxide, and they have a spherical shape. They have four polypeptide chains, and they have hydrophilic and hydrophobic parts. The hydrophilic pointing outwards. But why? The outward pointing hydrophilic groups on the surface of the molecule are essential in maintaining its solubility. And sickle cell anemia is an inherited blood disorder caused by substituting one amino acid, valine, for a normal amino acid, glutamine. And the problem that we will have is that the hemoglobin will be less soluble in the blood. Thanks for your time and your attention, and our videos will be shared as soon as possible. Thank you very much. Take care.